All right, brief review. What you need to know about key biological molecules and concepts, okay? First, perhaps most important biological molecule is water. So what do you need to know about water? You need to know two things. First, you need to understand the concept of hydrogen bonds. Look, this is the review, okay? I'm gonna ask this on the exam, but it's more of a review. So what is a hydrogen bond? Water molecule is a polar molecule. It's what's called a dipole, okay? It has slightly negatively charged oxygen, slightly positively charged hydrogen. So it turns out that positive and negative charges form a physical electromagnetic hydrogen bond between them. This is why water is liquid at the room temperature. Why it's important? Because hydrogen bonds hold various biological molecules together. Two strands of DNA are held together by hydrogen bonds. The shape of the protein is determined by hydrogen bonds. Hydrogen bonds play an very, very important role in biological systems. Second thing to know, what relates to water and solutions, is the concept of pH. So just remember that, look, I'm not going to ask you, it's not a chemistry class. I will not ask you to calculate pH. What I can ask you though, or what you should know, if I tell you that pH is neutral, it means that it is 7, okay? If it's acidic, less than 7. Basic, more than 7. And if I ask you, what happens to blah, blah, blah when pH decreases, you have to appreciate if it decreases, it means the system becomes more acidic. If pH increases, the system becomes more basic. Any questions on this part? Okay, good. That's going to be very simple, very straightforward. I'm kind of laying out the rules. So let's talk about some biological molecules. We're going to start with proteins, okay? So what do you need to know about the proteins? First of all, building blocks. Building blocks are amino acids, okay? Now, I have a privilege over you. That was tongue-in-cheek comment. I can erase things. Once you're writing, you really can't, okay? So I'm going to be using that advantage to myself. So this is the formula of amino acid. Is it a chemistry class? No. So you will not be asked to write a formula of amino acid or to recognize it. But I need it for the illustration purposes. Okay, so this is your amino acid with a side chain. Okay, we good? Now what if you take this amino acid and you put another amino acid next to it, and you run a reaction of dehydration, which will be, so water goes out and carbonyl group and amido group form a bond, which is called a peptide bond. Now look, you have two amino acids. Can you see that? Two amino acid residues connected by a peptide bond. You can slap another residue over here or another residue over here, and you can extend the polypeptide, the peptide chain. Does that make sense? So you build it, and every synthesis reaction in this case, addition of each amino acid is the reaction of dehydration, which, by the way, means that if you need to break the chain in pieces, that will be a reaction of hydrolysis. So anyway, you make the chain, and you have those side chains of of amino acids sticking out. These side chains are different. There are 21 different amino acids that can be found in proteins, in the living things. So these different chains can be polar, can be non-polar, can be positively or negatively charged, can be bulky, can be small. So they will be interacting with each other. Does that make sense? So these side chains will force the polypeptide to start folding. Are you with me? I am a sucker for analogies. I cannot create the good analogy for this one. Like, I can't possibly do that, okay? So basically, the polypeptide chain just cannot exist as a straight chain. So what do we have here? Polypeptide chain is called a primary structure of the protein. So primary structure is the polypeptide, this sequence of amino acids. Then this polypeptide starts folding into secondary structure, okay, which is composed of two key elements, alpha helices and beta sheets. A little illustration how complex this is. So right now we have enough computing power to predict whether a certain stretch of amino acids will fold into the alpha helix or beta sheet. Does that make sense? Like, now we can do that. Earth as a whole does not have enough computing power to predict the folding of the entire protein structure into the tertiary structure. So tertiary structure, I usually refer to it as a globule. So what happens during folding? Now, let me kind of pause writing for a second. So first of all, these carbonyl groups, these amido groups, they will form hydrogen bonds. Good? Side chains will interact with each other. They will interact through polar-polar interactions, or positive-negative charges, or dispersion forces between the hydrophobic residues. The point is, they will interact and they will force the polypeptide to fold, first in alpha helices and beta sheets, and then alpha helices and beta sheets will fold further. For many proteins, this globular structure is it. Good example, spike protein of a coronavirus. It's a single polypeptide which folds and forms a protein. Does that make sense? For some of the proteins, tertiary structure is not the terminal step. They form quaternary structure, which consists of several globules, which basically means several individual folded polypeptides. Okay, so a good example of this would be RNA polymerase of bacteria, E. coli, which consists of 5 subunits. At this point, I used to bring up the reference to a Japanese cartoon called Voltron, but it only signifies how old I am because nobody else knows. Well, somebody knows what Voltron is? Okay. It's basically a giant robot that is composed of 5 smaller vehicles. So you can isolate, in case of RNA polymerase of E. coli, there are 5 subunits. You can individually isolate each subunit and keep it in a vial. But in order for the enzyme to work, you have to take 5 subunits and put them all together, and they will form a certain structure that will be functional. This is called quaternary structure. Are we clear? Why I pay so much attention to this? If we manage to change an amino acid, we manage, possibly, to change the structures, because side chains will determine the structure. See what I'm saying? We replace the amino acid, we replace the side chain, we possibly altering the structure, which means we possibly altering the function. Last year there was a manuscript about influenza virus demonstrating that you need just one amino acid change. I mean, for instance, if you change that particular amino acid, it allows the virus to significantly increase its ability to enter the cells. Just one, which is remarkable. Like, it changes the function of the whole structure, not just one protein, the whole virus. You see why it's important? Now, what is the function of the proteins? I usually like to think about proteins as molecules that do stuff. So they are structure, they are enzymes, signaling. Okay? Now, it's not like 100% of all these functions are carried out by proteins, but think of it this way. If there is some function in a cell, there is a pretty good chance that this function will be carried out by proteins. So you can sometimes think of the proteins as things that actually carry out the phenotype, which we'll talk about. Any questions about proteins? Okay. Let's do carbohydrates. So carbohydrates consist of monosaccharides, and just like proteins, you have this fancy reaction. So you have monosaccharide plus monosaccharide plus monosaccharide, and eventually, like, you slap them one on another, you get a polysaccharide. Okay? So this is the reaction like any other reaction of synthesis. It's the reaction of dehydration. Okay? Now, you will see today that this is like important to understand, that you slap many monosaccharides forming a long polysaccharide chain. In order to break it down, you need to add water, lysis, right? What are the functions of carbohydrates? Structure. Turns out fungal and bacterial cell walls, they are composed of different polysaccharides, but those are polysaccharides. And of course, energy. Okay. Now let's do lipids. Okay, and we're going to put lipids right over here. So among lipids, we want you to know three types of molecules. The first one would be triglycerides. So these are energy-bearing molecules. There's no other function assigned to them, really. So here's what they look like. So you have glycerol residue, okay, here. That's the glycerol. And each glycerol is connected to a molecule of a fatty acid, a really, really hydrophobic molecule. Now this chain, zigzag chain behind carbon represents just a fatty tail. If you're familiar with organic chemistry, fine. If not, don't feel left out. The point is, this whole thing is A, superiorly hydrophobic, B, bears a lot of energy. Does that make sense? Now, second category that I want you to know are phospholipids, sometimes referred to as phosphoglycerides. So a typical phospholipid, I'm going to orient it differently, also carries glycerol, okay? But this time, this glycerol is attached to only two fatty acids, and importantly, one of them is usually unsaturated, but that's okay, it's not super important. What is important, really, really important, and I forgot oxygen here. Okay. What really is important, there is a phosphate group right over here. Okay? There's a phosphate. Phosphate is the inorganic anion, which is negatively charged. Okay? And this part of phospholipid molecule right here is polar. Or sometimes we refer to this molecule, to its chemical properties, as being hydrophilic. Okay? Now this part here is non-polar, or as we refer to it, being hydrophobic. Okay, does that make sense? This is called amphiphilic nature. Amphi means both. So the molecule of phospholipid contains both hydrophilic and the hydrophobic part, which explains why phospholipids can form a cell membrane that looks like this, where hydrophilic heads are facing either the inside of the cell, the cytoplasm, or the outside of the cell. Okay, again, watery environment. And at the same time, the inside of the membrane, that meat in the sandwich, is hydrophobic. And it determines the semi-permeability of the membrane that can let only hydrophobic, non-polar compounds in directly. The polar have to find a different way. Okay? Now, when I told you that the polar head is negatively charged, you know, that's important. The fact that the surface of the membrane is important, that the surface of the membrane is negatively charged, it's going to become very, very useful, that knowledge, when we're going to talk about various antimicrobial chemicals that we use in a household environment or in a hospital environment. Right? Good? Question so far? Okay. Now, last little bit that I'm going to slap here. No formulas, I promise. Also lipids, hopanoids, and sterols. Okay? So hopanoids are really complex lipids, many fused cycles, which are found in prokaryotes or bacteria. Sterols are found in eukaryotes. So what is the function of these two? They regulate the fluidity, or in other words, rigidity of a membrane. Okay, so the structure of the membrane is important for the transmembrane transport. Okay, so sterols and hopanoids, by being introduced into the membrane, can change the transport features, the structural features of the membrane. Are we clear? Okay, both of them do the same job. They just, hopanoids are in bacteria and sterols are eukaryotes. Questions here? Good? Okay. Last biological molecule that we need to talk about, nucleic acids. Okay. So, DNA. Double-stranded. Genetic information storage. Okay. RNA. Most frequently, single-stranded. Not always. Genetic information transport. Again, we kind of are building the story here, okay? So it's going to get a little bit more complicated than that. Now, any questions on this part? We're going to talk a lot about this in the Unit 2. Okay, now, this course will be built and is built on the understanding of two fundamental biological concepts. One biological concept is evolution. Evolution happens, we can talk like an hour about evolution just in biological systems, but what I want you to understand is evolution happens under selective pressure, okay? Does that make sense? So there are several good examples of the evolution happening. And I'm going to try to provide a relevant example and what can be more relevant than coronavirus, okay? So, does it evolve? Yes. Does it experience selective pressure? Yes. Does it evolve to become more transmissible? No, because there is no selective pressure to become more transmissible. It's pretty darn well transmissible, okay? What evolution is directed towards is better fitness. Does the virus become more fit? Most likely, yes. What kind of selective pressure we put on the virus? Well, vaccines, antibodies, we put that selective pressure, okay? Can it completely avoid? Turns out, no. It can avoid some of the selective pressure, but not all of it. You see what I'm saying? So when you think about a certain quality, a certain phenotype, and we'll get to phenotype in a second, a certain phenotype, and you ask yourself, how did that happen? You have to think, okay, what kind of selective pressure this population of organisms could have experienced over, certain evolutionary period. Does that make sense? Okay. Second, really, really important concept, the concept of gene expression. And I'm going to do it in a classic way. You probably know that way. So we've got DNA that is transcribed, so that is transcription, into RNA, which is translated into a protein. Now let me spend a few minutes here addressing this idea. I think I came up with a pretty good analogy. So, what is DNA? Let's take bacteria E. coli, okay? DNA is a recipe book, a family recipe book, which contains all the recipes ever created in the family. So E. coli's recipe book contains 5,000 recipes, 5,000 genes, okay? When friends come over, do you cook the entire book of recipes? No, never, right? You pick some that will suit the occasion, like, one of your friends is vegetarian, so you've got to have a vegetarian option, another one has celiac, so you got to have gluten-free, and so on and so forth, okay? So you pick 5, 6, 7 maybe dishes, and you cook them. But before you cook them, you have to transfer those recipes to the kitchen. Not going to carry the entire book with all 5,000 recipes to the kitchen. You need some kind of a transport molecule, okay? So what you do, you take your phone and you snap the pictures of the recipes. That's those pictures of the recipes. It's your RNA. See what I'm saying? Now, you look at the pictures and you cook the dishes. Kitchen, by the way, in this case is a ribosome, okay? So you've got the dishes, and your guests are going to be eating the dishes. They're not going to be eating the photographs. They're not going to be eating the recipes on the paper. They want the dish. So in the cell wants the dish, the stuff that they do things. I'm not saying that RNA doesn't. There are some molecules, RNA molecules that have enzymatic activity, but that's beyond the point. My point is, First of all, if you have a gene, you're going to have a protein and vice versa. Second, the phenotype of a cell is not defined by all the genes. It is defined by the genes that are produced at the given moment. Does that make sense? And these genes can change, and they can change in response to the environment. You see what I'm saying? So this is really important to understand that E. coli does not produce 5,000 genes. It produces certain genes at a given moment. And since we're going to be focusing on clinically relevant stuff, we're going to be talking about antibiotic resistance, toxins, virulence, various virulence factors. Every time we say, oh, this bacteria have flagellum, which means they can swim and they become more spreadable, more virulent. You have to think, haha, flagellum, where does that come from? Well, it turns out that bacteria have a cluster of genes that allow these bacteria to make flagellum. And if you take away that cluster of genes, they can't make flagellum. So it's all in the DNA. Does that make sense? Any questions? Okay.